Yeah, go ahead. Yeah.
And here you see a picture of me. I guess the only thing of interest here is that when I started working on PiMS, I had salt and pepper hair, a bit like George Clooney, and as you see, it's been a challenging 8 years. So what I'd like to talk about is why you might be considering using a LIMS for protein production, something about the people who've used PiMS and what they have to say about it, and then get into the 5 basic concepts that a PiMS user needs to be on top of. I'll talk a bit also about an extension for crystallogenesis and something about our future plans. So why might you want to use a LIMS in your research work? Well, to start with, have you ever had the experience of opening up a freezer and taking out an Eppendorf and not being able to find out what's in it and discarding it? At that point, you've thrown away the cost of the reagents that we use to produce that. More importantly, you've discarded a scientist's time, and you've registered that some knowledge has been lost. Or have you ever had difficulty expressing a protein and thought, well, we had a problem like this 2 years ago, and we managed to solve it, and then gone to the bookshelf and tried to find the right lab notebook to look in? I know of one laboratory that uses PiMS where the professor logs on every evening to find out what's happened in the lab. One postdoc I know told me that at the beginning of her second postdoc contract, they pointed to the drawer in the freezer where all her DNA samples were and handed over the lab notebook of her predecessor. So she opened it up, and it was written in Thai. One laboratory which uses PiMS told me they did an audit of their primer orders from the year before they adopted PiMS and found that they'd wasted $1,500 because of mistakes cutting and pasting between different pieces of software. So in face of these challenges, a number of labs have decided that they would rather record their information electronically. And if you look at your current workflow, it's relatively easy to identify a series of forms that need to be filled in and to design a series of database tables. And so a number of people have made a FileMaker application to do that. However, there's a problem. A LIMS developed that way doesn't seem to transfer to a second laboratory. I believe, in fact, that PiMS is the only LIMS in use in more than 3 protein production laboratories.
Chris, just for a second, I think, are you on the speakerphone or something? Before, I think we had a better quality. It's just a little bit of the kind of.
Okay. Is this better now?
I think it's slightly better right now. Yeah.
Yeah. Okay. We were fortunate in the development of PiMS that right from the beginning we were charged with supporting 2 different research collaborations. So we had to build in flexibility, and it's proved that makes PiMS not only flexible for different laboratories now, but flexible for a single laboratory as its methods change. So a LIMS is especially suitable if you're doing high throughput work, if your projects are long term, if you collaborate with people off-site in other organizations, if some of your work is by robotic instruments, and also if you're working on a service basis, so you need to report back to external clients. Now, there are 2 buzzwords that are used in this field. One is LIMS and the other is ELN, or Electronic Lab Notebook. What's the difference? This refers to a continuum. At the one extreme, actually, the electronic recording that's most used is Microsoft Office. And it's very easy to type data into into Word, it's a great deal less easy to search after you've done that. Similarly, Contour ELN makes data entry fairly free format, and some people have repurposed a wiki for recording data. Then at the middle of the range, you can take on a general purpose LIMS that understands the notion of a sample and experimental procedure. As a matter of fact, a customer relationship management system is also fairly good for tracking progress of projects. And then at the other end of the range, there's software that actually understands the structure of molecular biology information. At the beginning of the pipeline, Vector NCI, at the end of it, PDB, and PiMS spans the range from the selected target to the diffraction-ready sample. There's a great deal of discussion now about the crisis of drug discovery, that somehow the enormous increases in the productivity of research biologists are not being reflected in an increase in the rate of drug discovery. On the contrary, it's declining despite increase in spending. There's lots of different approaches to this under discussion, but all of them seem to me to have one thing in common. That they involve collaborations between a range of different organizations. No one imagines that successful drug discovery happens in-house in a single company. And the result of that is that there's a need for good ways of sharing knowledge across organizations. Now, there is some downside with a LIMS. It needs to be backed up reliably, so there's some systems administration overhead. For scientists in Europe, I have funding from Instruct to provide a software as a service version of LIMS, and they can simply email me and ask for a login for academic purposes across Europe. I'm pleased to say that SBGrid is considering providing the same service. And if there's a PiMS user community in the American time zone, I think that will make working together on adapting it for a variety of research purposes quite effective. A LIMS isn't so useful if someone halfway along the pipeline doesn't enter records of what they're doing, then the chain of custody is lost. And one of the challenges here is that the cost of entering data happens today as you're doing the experiment, and the benefits come in 6 months or 2 years' time when you went to look back at it. Unfortunately, the human species doesn't respond very well to that sort of challenge. So what are the costs associated with data management? If you're doing it the traditional way with a paper lab notebook, then the cost of setup is very low and the cost of entering the data is low. The cost of retrieval is higher, and I would suggest that the most significant cost of all is the cost of lost data. If you're using MS Office, then the cost of setup and entry becomes a little higher, and the cost of retrieval and loss becomes a little lower. And as you move in a more formal direction, so the cost of setup and entry increase, and the cost of retrieval and loss get lower. But in PiMS, because it understands typical working methods in molecular biology and protein purification, we have been able to reduce the cost of setup again, and we work hard to make data entry as user-friendly as we can. So who uses PiMS? We were first funded to support the Membrane Protein Structure Initiative, a collaboration of a dozen UK laboratories, and the Scottish Structural Proteomics Facility, also in the UK. And this tells you the number of records they created. Then in 2007, the Oxford Protein Production Facility turned off its previous commercial LIMS, ceased entering data to be exact, in its previous commercial LIMS, and adopted PiMS. and it's been using it ever since. These counts of records, I'm afraid, are rather out of date. In 2012, PiMS was adopted at a variety of universities, including the ones listed on the slide. And the Extel PiMS extension was adopted a couple of years ago in Aulu and has been in use in the OPPF for some while. PiMS is free for academic use and always will be. For commercial use, we have recently concluded a partnership with Emerald Bio, and you're welcome to contact them for details of how to use it. That agreement is very new, and I'm afraid we're still in process of rewriting the academic license. That's meant that we couldn't conclude the discussion with SBGrid about licensing them to provide a software as a service version. That delay is wholly on our end, but I hope to finish up that discussion in January. So a couple of years ago, we did a survey of, at the time there were 18 academic licensees of PiMS, and there was a high response rate, and as you can see, the reaction is best described as mixed. One person was kind enough to say, I love PiMS, and there were 4 other comments that were equally positive. There were also 4 comments right at the other end of the spectrum, equally negative, that the learning curve was too steep for us, that there was an energy barrier to using it. Now, clearly, there's some challenges here for us, for those of us who develop PiMS, to reduce the size of that energy barrier. If I may say so, I think there's also a challenge to scientists here, that the community does appear to be changing its mind as its work rate gets higher and higher with the assistance of better reagents and instruments. There's also recognized to be a greater need for electronic record keeping to keep track and to get the full benefit from the records. If you're interested in the techie bit, then PiMS is a web application. You simply log on to it. We test it in Internet Explorer, in Firefox, in Safari, in Chrome, and the new release now, most of the functionalities also work on Android and on iPhones. iPads, sorry. We haven't adapted it for the really small screen of the iPhone. That's possible future work. It uses a fairly standard range of open source software at the server side. So the features in the forthcoming release, PiMS support works on open reading frames, also on complexes. One person is interested not actually in the protein that's expressed, but in the promoter. Some people use it with protein from natural source. Because it can be customized in a way I'm going to show you, it could be used with Gateway or Infusion or indeed any workflow. It has support for site-directed mutagenesis with a wizard for designing quick change primers. It supports individual experiments and also work with plates. There's recently a freezer view to help you track your sample, and will soon be a facility for booking an instrument. So you can say, tomorrow afternoon I'd like to use the actors. Another important variable in this software is who can read the data that's entered. If you're typing your data into a Word document, then it's essentially private. Sharing the data involves some effort and inconvenience. If you've installed a wiki, then you're making your data public to anyone within your firewall. In between those cases is the more complicated one, where you can manage the access, and that's what PiMS offers. If you're a single tightly knit research group, then you probably simply have a single lab notebook which all the PiMS pages are part of. But if you've got external clients, then you probably have one for each of them, so you can give them a read-only login to access them. So the key concepts in PiMS, which you need to understand how PiMS uses these terms to begin using it. It's a target, that's to say the open reading frame of interest. A complex, if you're interested in several targets that you believe to be complex together in vivo. The construct, that's to say your plan for expressing it, the truncation, the mutagenesis, details of the vector that you have in mind. Once you've recorded the construct, you're ready to start recording laboratory activity. For each sample on record, PiMS knows its type. So if you've said that this is soluble protein, and you ask what's the next experiment I can do with this, you'll get a sensible list that includes concentration, but doesn't include PCR. And an experiment is an activity in the lab. It makes use of some samples and it produces some new ones. A protocol is a template for an experiment that says what information you need to decide as you plan it and what observations you're going to record. So let's have a look at, so here's a diagram of how this joins together. And then let's have a look at some screenshots. Here's some details of a target. You don't have to type all this in because PiMS is capable of downloading this information from a public database. So that's how these sequences were obtained. Because the database is linked together, then low down this page, actually, there's links to quite a lot of information about this target. Here you'll see the details of a construct, in this particular case a full-length one, with the one of the boxes is open up that shows you details of the reverse primer. PiMS includes a wizard for designing the primers. Here's a record of a sample, in this particular case a sample of plasmid, with some details of the experiment which produced it. Here's a record of an experiment, in this particular case a PCR. You'll see the setup parameters, the decisions made when planning it. Now, you might always use the same number of cycles, so you might not want to prompt your scientist to enter that every time. So in that case, you want a custom protocol. Here is a PiMS protocol, in fact one for bicistronic infusion, so the input samples it has are two different PCR products, as well as a vector. So when you tell PiMS you're doing an experiment with this protocol, it'll prompt you for the two PCR products that you're going to use. This is a diagram of a workflow. It's not a planned workflow, it's an actual one. In fact, it's one that resulted in a publication in Nature, the structure of the hedgehog complex. So the diamond at the top represents the hypothesized complex. Below that you see the two open reading frames. And an oval on this represents an experiment. There were two PCRs followed by digest and ligation. And then in the left-hand branch, the purification process was relatively simple. In the right-hand branch, it proved to be much more challenging. But then finally, the two join up in a complexation experiment that was followed by another purification, a concentration, and crystal trials. And this particular report was obtained after the first crystal hit. So those details are also available as a report as a PDF, and this is a copy of a PDF produced by PiMS. And if you could see the whole page, you'd see the whole history. So that's the information that you'd want in the method section of the paper. It includes gel images that you've uploaded as you've done the experiments. Another use for it, you could print it out, sign it, and stick it in your lab notebook, and then you've got a record of priority. You could also send it to an external client who you've been doing that work for. Here's the front page of Extal PiMS. This can be connected to a formulatrix instrument or the obsolete Rhombix instrument. I would like to support Rigaku equipment too, but that work hasn't been done yet. And here you see the image of a well. If you click that green arrow over here, it'll whiz through the well images, and there's these handy shortcuts. For instance, if you press space at a well of interest, it'll pause. I know someone who got a bit behind and needed to review 10,000 well images before he went on holiday, and he did in fact succeed thanks to these handy shortcuts. It'll also animate a movie showing the history of a well. That's quite useful if there's something that looks like a crystal. If you go back to the start and see an air bubble there, then you'll know that unfortunately it's not. In Oxford, they've also recorded some tearjerker movies where you can see a crystal forming in the well and then, I'm afraid, dissolving again. So the plans for next year's work include a bigger range of reports to support the lab manager, better facilities for user management. I'm afraid if SB grids do agree to support it, they've got some inconvenience until I've done that. It's a little clumsy at the moment to add new users. Following that, we've got some planned improvements on the usability of PiMS from the point of view of the experimenters. There's some investment significant investment by Emerald Biosystems, and in addition, a number of academic contributions, an EU grant. Our university has just contributed code to record crystal treatments before diffraction, and CCP4 is funding me to give user support to academic users. Beyond that, we'd like to work on support for mobile clients to give it a richer model of DNA chemistry, to support reagent ordering, and Oxford University's group there has applied for funds to integrate some bioinformatics to support construct design. The productivity of structural biologists has increased by a couple of orders of magnitude in the last 2 decades, and I don't know any other trade that's accomplished that. That's partly because of better reagents, it's partly because of better instruments, it's partly because they're hard-working people, it's partly also because the software available to them has improved. And I think what structural biology now needs and deserves is a consistent user interface all the way from target selection to interpretation of the structure, with seamless data transfer all the way along that line. And that's the vision that this work is once more part of. On this final slide, you can see the PIs who guided the work, the developers who contributed, and I'd most particularly like to thank all those scientists who've come to me and said, look, what it does isn't quite right on this page, I'd like it to do that. So now it would be great if there are some comments and also some questions.
Thank you, Chris. This was excellent, and there's a number of questions here coming up from users. First one is, in terms of the ordering database, some labs like to keep the database of everything that has been ordered and potentially accumulate simulate them before contacting vendors. Is this something that is supported through the software and through the LIMS as well, or not?
Hi, I'm afraid I can't hear what's being said. I can just hear that someone's saying something, but I do have a question.
Okay, sorry. Yeah.
Through the chat route.
I'll repeat it. Can you hear me okay now?
Yeah, I can. Thank you.
Yeah, I think my microphone was off. In terms of some labs are keeping ordering databases whenever all the chemicals, everything that goes to vendors, it's included in this table of database. And do you have any functionality for this within LIMS?
We have reference data of reagents that are available, but we don't connect that to the forms that vendors provide.
Okay, but just in terms of just even keeping a list of everything that was ordered within the last year, or entering things as they're being ordered, that's not something.
Yes, if you when you no, we don't record orders. You can use it to record reagent stocks.
I see.
That's particularly valuable if you want to track batch numbers. For example, I know one group that does its eukaryotic expression with fetal bovine serum, and that's highly unreliable. So if you run into problems in one experiment, you may want to go and have another look at the other experiments that were done with the same batch.
Okay.
There's similar issues with anything that's got a shelf life, and in that case, you want to track the individual bottles. If the reagent is sodium chloride, then you can pretty much rely on the supplier that it will be sodium chloride in the bottle, and there's less value in trying to have a very disciplined approach with that.
Another question from a participant here. So the question is if there is any, maybe now or in the future, ability to upload X-ray data sets, diffraction data sets?
Yes. Yes, you can do that. We've got a protocol for X-ray experiments, and you can, like with any other experiment, you can upload files. In addition, we're discussing with the UK synchrotron, the Diamond Light Source, a facility to automatically download the diffraction results from Diamond. What we're most likely to do is, well, we're most likely to save a link to the images because they are very big nowadays. I know one group which took home a terabyte of data after a 16-hour session. What Diamond do, and some other synchrotrons, is they have an automated processing pipeline, so you can actually get the reduced data from them. And most of the time, people consider the reduction acceptable. Maybe one time in 5, when you look at it, you say, actually, I can do better than that, and then you do have to go and get the raw data.
In terms of data security, because you're talking about the data sharing and having those electronic notebooks shared with individual people in the lab, etc., how secure is it behind the scenes in terms of doing encryptions, other things, or did you guys plan for that as well?
Yes, we did. Part of the release procedure is fuzz testing. So it's tested. It's designed and tested to be secure against HTML injection and SQL injection, and there's an optional defense against cross-site request forgery. That's an installation option. It imposes a certain amount of inconvenience to the user, so that it's an installation decision whether you turn that on or not. In terms of encryption, you can configure it to use HTTPS for the connection. And if you don't trust the hosting provider, you can arrange that the database is stored on an encrypted drive.
You also mentioned supported clients, that there are standalone clients and you can use the web. So you guys have 12 users, 12 major users right now. You mentioned which ones are more they prefer to use.
Oh, no, there's not a remote client software. You simply use a browser.
Oh, browser. So it's everything is done through the browser. Okay.
Yeah, yeah.
Okay. Okay. So do you guys, if there are any other questions from participants, you can send them to me right now. Otherwise, we'll be concluding. I mean, it sounds all very, very interesting. I hope we can deploy it fairly soon through SB Grid, and I know my lab at Harvard Medical School will be certainly interested in testing it and putting it to use in our